I'm here at ASH, and yesterday my research group presented the latest results of our research activity. So we focus on multiple myeloma, and in multiple myeloma we specifically look at the genome of the cancer cells to understand what are the pathological bases of disease initiation and disease progression. We use that using next generation sequencing technologies, and while in the previous years we focused especially on analysis of the coding genome, which means analysis of mutated genes and their possible relevance to cancer, lately we've been focusing on the analysis of the whole of the genome, which means the genes themselves plus the 98% of DNA sequences that actually do not code for proteins. Of course, that's the vast majority of the genetic material of the cell, and in fact in multiple myeloma we found a huge number of somatic alterations that are alterations acquired by the cancer cell at the time of transformation or after. These alterations are in the form of point mutations, of course, but what's even more interesting are what we call structural variants. So these would be events whereby chunks of DNA get either deleted or amplified or translocated to other chromosomes or inverted or duplicated. So of course these are events that play a possible role in disease pathogenesis because they do not code for proteins, but they may alter the way proteins are expressed. So in our work we did go to the root of why some of these events happen, and we found some signatures of what we call mutational processes that are operative and may explain why such structural rearrangements arise in the first place. We went on to describe the main types of these rearrangements, and myeloma has a vast array that is not as common for hematological malignancies. One could say that because of these structural variants, myeloma looks more like a solid cancer from a genomic point of view than a hematological cancer. We then went on to understand whether these events have a role in dysregulation of known oncogenes, so proteins that may have a causal role in cancer, and in fact we found that contrary to what previously known, some oncogenes are targeted by these complex rearrangements way more often than they are by classical translocations which are routinely picked up by fish in patient samples. So our results open a whole new field of investigation in multiple myeloma that of course will require hard work of validation of these events as effectively pathogenic in cancer, but that certainly furthers our understanding of the mechanisms leading to disease initiation and progression, and hopefully in the future will give us a better way to track the disease and possibly to target some of the key features.